Okay, so in humans, 90% of the information coming from the eyes, right, leaving from the ganglion cell axons that form the optic nerves on the left and right, are going to work their way back to the thalamus, which is that set of nuclei that sit above the brain stem. So I'm going to take the brain apart here, and I'm going to remove the cerebellum. And then come over here, and you can sort of see here's the brain stem, and the thalamus is this big structure. It's going to move its way. Its medial structure is in the middle, but it sort of extends from the midline, to the side on either side. A big sort of egg-shaped, set of nuclei that are, processing independent afferent channels of input, like somatosensory touch, for example, like visceral sensations, like auditory information, like gustatory information. Actually, every kind of incoming afferent, channel, sensory channel, and except for olfaction, except for smell. And then the thalamus, there's individual nuclei in the thalamus that are processing sound or, light or, touch, etc., are going to then send their axonal projections to very specific areas of cortex, which are going to be initial arrival sites for, sensory information at the level of the cortex. There's also tremendous amounts of feedback, into these specific thalamic nuclei. So 90% of the visual information from the eye that's working its way back, into the brain is going to make a stop in a nucleus in the thalamus called the lateral geniculate nucleus. And it's called that because the thalamus it looked to early anatomists like it was sort of sitting on these little bumps, these little what they refer to as these little knees, like it's sort of kneeling. It looked like the thalamus was kneeling, and there was a bump or a knee that was towards the middle, so they called that the medial geniculate. Genu is Latin for knee, and geniculate means little knee. So there's the medial geniculate nucleus that, it turns out, actually processes auditory information before sending it on to areas in the temporal lobe of the cortex. While the lateral geniculate, the knee, to the side, on this side it would be over to here, on this side it would be over to there, was essential for the initial processing of visual information from the eye. 90% of that visual information in humans. And from there, the axons of the lateral geniculate nucleus, which is often referred to as the LGN, which stands for lateral geniculate nucleus, sort of radiate out back to the occipital lobe. There's this thing called the calcerine fissure. So right around this region in the midline here and towards the very tip back here is what we call primary visual cortex, which is the initial arrival site for about 90% of the visual information coming from the eyes. And this is going to be really, really critically important for sort of conscious visual perception, is this 90% that's working its way back into cortex. However, there are other targets of visual projections from the eye. And for example, about 7% of the cabling sort of actually swings over to the back of the brainstem to a set of structures up here. There's these little bumps. There's what we call the superior bumps and then the inferior bumps. And the superior bumps, the Latin for bump is colliculus. So these are called the superior colliculi. And they're really important for processing sort of visual reflexive responses. For example, if somebody throws something at you quickly, you turn and you quickly sort of deflect, right, in the right direction. If something unusual happened visually, you would orient, to what it was. So the superior colliculi are critically important for sort of visual orienting responses. And often these are rather, reflexive. They just like something's coming from here and you turn there. And they're also rather unconscious. You can, deploy, visual attention on something, but often they're quick to help you, navigate and to get around your environment sort of successfully. And then there's an area called the pretectal region, which is in the brain stem, that's receiving, another 1.5% or so of the projections. And this is a really important reflexive response that is involved in pupillary dilation and constriction. So basically, adjusting the size of the pupil in response to ambient light levels so that you're not, allowing in too much electromagnetic radiation so that, you're shutting down, basically all the photoreceptors and you're not getting any kind of visual discriminative ability, right? You want to get to the right aperture, the right opening to allow, so some of the photoreceptors are responding and some are not, right? So you have some information. It's information rich, the input that's allowed through the pupil. And then there are, remember, the intrinsically photosensitive retinal ganglion cells. These are actually in the ganglion cell layer of the retina, not the photoreceptive layer, but some of the ganglion cells actually express a pigment that can absorb, mostly towards the short or blue end of the spectrum. And they help to set circadian cycles. So they project back into the hypothalamus, which is a little bit below and anterior to the thalamus. And they project specifically to a nucleus that sits right above the chiasm where the optic nerves cross. This is called the supra, which means above, chiasmatic, above the chiasm nucleus. And those nuclei, those neurons in the SCN and the hypothalamus project back to a gland that sits on the back of the brainstem between all the colliculi. That's called the pineal gland. And remember, when light strikes the intrinsically photosensitive ganglion cells in the retina, it signals the suprachiasmatic nucleus, the hypothalamus, to tell the pineal gland to stop releasing a hormone called melatonin. And when light levels are dropping and there's less, particularly blue light stimulation of these intrinsically photosensitive ganglion cells, well, that signals the hypothalamus to allow release, to promote the release of this hormone called melatonin, which is a blood-borne signal that helps to, help you drift off, and start getting ready for sleep. So I think it's interesting that, 90% of the projections are going to thalamus, to the LGN, and then to the primary visual cortex in the occipital lobe in humans. And yet we do have, significant visual reflexive inputs and responses that are, accomplished via these other subcortical structures like the superior colliculi, the pretectal region of the brainstem, and the hypothalamus. In patients who have damage, back here to the primary visual cortex, this 90% of projections will report that they are blind, that they do not consciously perceive anything visually. And yet, if they will still maintain these other subcortical reflexive networks. And so if you toss a Nerf ball, towards somebody in that circumstance, they turn and deflect. So, and think of this, like other organisms have different kind of ratio in terms of the projections from their eyes, how much is going to these apparently more conscious, kinds of cortically mediated visual pathways versus these subcortical reflexive pathways. Like a cat, about 50% is actually projecting, from their eyes back to the superior colliculi. So cats are like, very quick to respond to things that are moving. And, whether they are actually, specifically visually conscious or just reflexively responding, to basically go after prey, for example, is an interesting question.